Gene editing is an astonishing technology. Picture a doctor curing genetic diseases by fixing a single faulty letter in a DNA code. It's kind of like deleting a sentence on a Google Doc. See those 5 letters? That's my brown hair. Alright? And when I delete that and change it with 5 new letters, I suddenly have blonde hair now instead of brown. That's kind of weird, isn't it? Now consider the excitement and anxiety that comes with the power to rewrite the genetics of living things. Gene editing began decades ago as a little more than a dream in laboratories, but recent breakthroughs, especially the invention of CRISPR, have turned that dream into a reality. In the last 10 years, things have accelerated at a pace that no one could have expected. Now labs across the globe, they have tools to cut, replace, or add DNA with precision thought impossible 10 years ago. And every day now, it brings new possibilities from fighting diseases to resurrecting extinct species using gene editing.
But power comes with questions. As gene editing becomes faster, cheaper, and more accurate, society must wrestle with its ethical dimensions. As we are ready for a future where babies can be designed to avoid illness or to have different traits from birth, who decides what's acceptable to change? What's the risk, both known and unknown, when we tinker with the foundation of life itself? As we explore this technology, you will learn about 3 things: how gene editing works, a new technology called CRISPR, and the ethics involving gene editing. Together, these forces are setting the stage on one of the most important events of our time, about science, sociology, and what it means to be human. Gene editing is a very new and an innovative process, and to fully understand it, we must understand how it works. Gene editing is a method for making specific changes to the DNA of a cell or organism. This can be done using to add DNA, remove DNA, or just alter DNA in a genome. For humans, gene editing technologies can be used on somatic cells or non-hereditable cells. Examples include muscles, skin, and organs, or germline cells, which are hereditary cells. So basically all types of gene cells.
The first type of gene editing is somatic gene editing. It is used to make edits in the somatic cells to treat genetically inherited diseases and is already in some clinical tests. For example, it has already been used in treating HIV and sickle cell anemia. These edits are limited to only the current patient and do not pass down to the next generation. This would be useful in treating people who already have the disease and have problems. With somatic gene editing, it makes a permanent change in the genome. So instead of a temporary fix, which has to be unkempt and taken often, it would be a permanent fix. It would also limit or prevent damage in things already caused by the disease. It has already been used in some areas like blood disorders, eye disorders, and certain cancer treatments. Somatic gene editing is just one way gene editing overall is showing to be a very promising option for disease treatment in the near future.
The next way gene editing works and is changing the medical field is hereditary human genome editing. Hereditary human genome editing is editing the nuclear DNA or edits to the genetic material found inside the nucleus of a eukaryotic cell in a way that makes it hereditary across generations. Unlike somatic gene editing, so what would happen is a parent knows that they have a genetic disease, and they could take this, and then their kids could not have genetic gene editing. According to the National Library of Medicine in 2017, around 5 to 7% of the population have genetically inherited diseases. This means that this would be a sizable percent of the population and could help future generations with less disease. This is a graph that shows how many people have heard about gene editing, and as it shows that more and more people are learning about gene editing, and compared to a couple of years ago, this would be basically all, not at all.
Now you may be wondering, I know these types of gene editing, but how does it work? Well, gene editing can be placed in 2 different cell methods, called vivo and ex vivo methods. In vivo methods, gene editing components are delivered straight into the body to make edits within the cells, while ex vivo are cells removed from the body, edited, and put back into the body. This whole process can take a few days in a lab and maybe weeks to years in a patient. In conclusion, gene editing works in 2 ways: somatic gene editing and heritable genome editing. Each way is useful in different ways to show a great potential in being one of the most useful tools we have in modern medicine and changes millions of lives. Imagine being able to correct every single letter in a vast instruction manual, and that defines who we are. That's our DNA. Every cell in your body carries about 3 billion of these letters, and yet a single typo in this enormous code can cause devastating diseases like cystic fibrosis, muscular dystrophy, and sickle cell anemia. For decades, scientists have dreamed of finding a way to fix these genetic diseases with pinpoint accuracy, and then out of nowhere came CRISPR, a powerful, elegant, and almost unbelievable tool that can actually rewrite the code of life created in Zug, Switzerland.
So what exactly is CRISPR? So the word CRISPR actually stands for clustered, regular, interspaced, short palindromic repeats. That's a lot of words. So what it is it's a natural defense system first discovered in bacteria that protects them from viruses. So if a bacteria gets a virus and it survives that virus, it keeps a little piece of that virus's DNA stored in little fragments in the CRISPR region of that genome, sort of like a vaccine.
Later, if that same virus attacks again, the bacteria produces two key molecules, CRISPR RNA and a protein called Cas9. The CRISPR RNA, or guide RNA, carries the memory of a virus. It tells Cas9 what to look for. Cas9 then acts like a molecular scissors when the guide RNA finds the matching viral genetic code. If it looks on Cas9, cuts the viral pieces of DNA, neutralizing the threat before it can cause harm.
This natural system of viral memory fascinated scientists. They had never seen anything like this before. They realized that they could reprogram this system into recognizing viral DNA to recognizing any genetic sequences like the human DNA. It transformed editing genes and changed biology forever.
Let's walk through how CRISPR works in a laboratory setting with this video.
Of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species.
It starts with the design of custom-made RNA. In just a short stretch of RNA that matches the DNA sequence the researcher wants to edit, think of RNA as the address on a GPS. It's the code that tells CRISPR exactly where to go in the genome. Next, researchers guide that pair of RNA with Cas9 and the DNA cutting system together. Now they form the CRISPR-Cas9 complex.
When this complex is introduced to a cell, the guide RNA searches through billions of DNA letters until it finds the perfect match for the CRISPR-Cas9 complex. When it does, the Cas9 makes a double-stranded cut, essentially breaking that strand of DNA right at the site. Now the cell senses this damage and immediately activates its natural repair system. And here's where the true power of CRISPR lies.
Cells have two main ways to fix broken DNA. The first is called non-homologous and joining. It's quick but sloppy. The cell glues the DNA ends back together, often introducing tiny insertions or detections. Scientists can use this method to knock out or deactivate a gene. Second method is called homology, detected repair. This is one of, or more precise, versions of it. If a scientist can provide a new piece of DNA, like a correct version of a defective gene, the cell can use a template to repair or break seamlessly, swapping it in the corrected sequence. In this way, CRISPR doesn't just cut DNA, it can rewrite it.
Imagine using this to fix mutations responsible for sickle cell disease, where one letter change in the gene for hemoglobin distorts red blood cells In fact, that's no longer an imagination. Clinical trials have already shown that CRISPR can effectively cure this disease by editing a patient's own cells. Scientists extract this bone marrow cells, correct all the defective genes using CRISPR, and then return those repaired cells to the patient where they produce healthy blood cells for life.
And medicine is just one area. In agriculture, CRISPR can make crops that survive droughts and resist pests without chemical pesticides or grow more nutritious food, because CRISPR doesn't always introduce foreign DNA. It sidesteps many controversies surrounding older genetic modifications, says Perelman 2024. The same tool used to treat human diseases can help create more available and sustainable forests for food in the future. The story of CRISPR is still unfolding. What began as a survival bacterial mechanism has become one of humanity's most extraordinary inventions, a set of tools to read and rewrite and redesign the code of life. Like all powerful tools, its future to depend on the hands who use it. Used wisely, CRISPR can eliminate genetic diseases and revolutionize the way we think of life.
As we look at CRISPR, asks us for balance between curiosity and caution, because as it would happen, gene editing sounds great. I mean, like, who wouldn't want to cure sickle cell disease, terrible diseases like HIV, developmental disorders, and stuff like that? But where do we draw the line? Where do we say that gene editing has gone too far from helpful to unnecessary, like changing your hair color to blonde? This stems from the ethical concern of gene editing. So the first concern of gene editing is safety. Now, as the video said, there are millions of these tiny letters in the DNA, and as Robert showed us earlier, these letters are few, like there are so many and they're so small that you could make any accidental change, even with CRISPR, any gene could be messed up. And think of the consequences of that. These consequences could include developmental disorders and higher risk for these diseases that we were trying to cure, even a higher risk of forming cancer. These facts were acknowledged in both the International Summit on Human Gene Editing, a global discussion in 2015, and the Second International Gene Editing Summit in 2019. These summits discussed the concept of CRISPR and concluded, according to a report from that, the CRISPR-Cas9 system is still undergoing development to reach the level of safety where it could be used in clinical applications, as well as them saying, paraphrased, that they don't think that it would be wise, nor would it be responsible to instate these gene editing technologies just yet, as the world, as we just don't know enough about these things yet. Another concern about gene editing is the consent of the person whose genes are being edited. Now, they cannot actually give this. Gene editing happens in the embryo stage, so they have not fully formed into a human yet. You have to change their genes before they become a human, or you can't change them at all. So what if this person what if a parent wanted to give their child blonde hair, even though both parents have black hair, and they wanted to make sure their child got blonde hair, and then their child ended up with blonde hair and hated their blonde hair? What could the child do about that? There's nothing that they could have said or done to make sure that they could have had the better hair color in their opinion. And not only does this affect the child whose genes were edited, but it also affects their future generations, as if they have this gene for blonde hair, then they might have the chance of passing that gene on to their later generations, and they will also have that same problem that the first generation had.
Now let's just take a moment to just process that information. It's a lot to process. Now, there's also issues with funding, as there is no federal funding for gene editing because of the risk previously mentioned. According to What Are the Ethical Concerns of Gene Editing from the National Institute of Health, federal funds cannot be used for any of the research that create or destroy embryos. On top of all of these ethical concerns, funding for gene editing is not being distributed, as it would be very inaccessible to the general public. As to edit an embryo's genes, a parent would have to pay a fortune. These concerns are really the main reason that gene editing isn't a global normality. However, we think that it will be sometime soon, it can reach a point to where they can be resolved, and then research for gene editing will become much more common to see. Maybe there will be government funding, but certainly there's no telling where gene editing will go. Look at these guys' hair colors. Don't they look weird? Well, that's what gene editing can do. It stands at the intersection of science, ethics, and the future of humanity. Understanding how gene editing works beyond just CRISPR reveals the remarkable precision and potential of modern genetics to correct mutations and prevent devastating diseases. Yet, as with any powerful technology, its use demands careful thought. So I recommend, after this speech, just go and look at gene editing for a little bit. See what it can do.
The ethical questions surrounding gene editing asks us to consider not only what we can do, but if we really should do it. Balancing progress with responsibility will define how this technology shapes the society. CRISPR-Cas9, one of the most accessible and transformative tools, shows both a promise and the peril of rewriting life's code.
Thank you.
Thank you.